This is chapter 16, section 1 on population genetics and speciation. Section 1, section 1 is on genetic equilibrium. Yeah, there it is. By the time of Darwin's death, the idea of evolution by natural selection had gained wide acceptance among scientists. But with the birth of the field of genetics in the early 1900s, spurred by the rediscovery of Mendel's work on the mechanics of inheritance, many questions about evolution and natural selection resurfaced. So by the time Darwin died, everyone accepted evolution, but now that we have a new type of field of biology called genetics and inheritance, we can look at adaptation and natural selection through the eyes of genetics. Population genetics is the study of evolution from a genetic point of view, not just in general terms over like comparing fossils and saying these types of things changed, but actually looking at the inheritance of one population to the next. In a population, individuals vary, and so scientists can measure a trait in a large sample. A bell curve is basically the shape that many traits follow, meaning that the majority of a population is somewhere in the middle, and then you have outliers on either side. In this example, I'm using fish length. So there's a few fish that are long, a few fish that are short, but most of them are average. So if you plot the number of individuals versus length, you're going to end up with a nice bell curve. So what are the causes of variation within individuals in a species or population? There are environmental factors, for example, the amount or quality of food. There's also, of course, heredity. There are three main ways that variation can happen. That's mutation, recombination, and a random fusion of gametes. Just to review what these three things mean, because we've studied them already. A mutation is an error in copying genes. Recombination is the reassociation of genes. Recombination occurs during a crossing over in meiosis. That's when you have the two homologous chromosomes that pair up during a synapsis. And during synapsis, crossing over may occur, which is where you have parts of the chromosome which break off and reattach to its homologue. And then we have random fusion of gametes. There are hundreds of millions of sperm, but there's only one that actually fertilizes the egg. And which one out of those hundreds of million is pure chance? And remember, during meiosis, you have independent assortment and random, I forgot. My brain doesn't work on a Monday morning. Anyway, random assortment, independent, random, I don't remember, but you remember the two the two laws that came from Mendel. Why can't I remember it? Anyway, I'm just, okay, forget, move on. So anyway, we have out of all of the sperm that are made, all of them are genetically different because you have the chromosomes line up independent of each other and they dissociate. I don't remember what the word that they use specifically, but they separate randomly. Random separation, yeah, something like that. Anyway, by doing it this way and having evolved this way in sexual reproduction, it ensures that the offspring are not copies of the parents. And I've said it since the beginning in biology, is that genetic variation is the key to the reproductive success of a species. If we are all clones of each other, especially when you have something like the coronavirus that comes out, if it kills one, it kills all. So genetic variation is hugely important for the survival of a species, because if you have these random viruses that come out into the population, it affects all of us differently, so that there's a chance that some of us survive, some of us don't, but those who don't survive that particular virus will then have their genes taken out of the gene pool, which is the next topic. A gene pool is the total genetic information available in a population. So if you look at the frogs on the right-hand side, the gene pool consists of, what is that? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 12 green frogs, 2 purple and 1 red. Those are the genes that are available for that population. If you know the contents of the gene pool, you can apply rules based on probability to predict the expected genotypes and their frequencies in the next generation. And so this is the practical application of the concept of the gene pool. If you understand the genetics that are available in a population, you can use probability and you can predict the percentages of the genotypes for the next generation. So let's do an example. Suppose that there are 2 forms of an allele. We have capital A and lowercase a in a set of 10 gametes. If half of the gametes, which is 5, carry the allele big A, we would say that the allele frequency for big A is 0.5 or 50%. But in this case, we're going to use decimals. Allele frequency is determined by dividing the number of a certain allele by the total number of alleles of all types. It's just a straight up percentage. So phenotype frequency is equal to the number of individuals with a specific phenotype divided by the total number of individuals in a population. It's the same thing as genotype frequency, only, or allele frequency, only we're talking about phenotype, the way that it looks, its physical appearance, rather than the alleles. So the phenotype frequencies in the first generation, which you see in the first row, we have 50% of them are pink because we have 1, 2, 3, 4, 5, 6, 7, 8 individuals, and 4 of them, 1, 2, 3, 4 of them are pink. Remember, we're going according to phenotype, which is the physical appearance. So 0.5 of the first generation are pink. 0.5 of them, oh gosh, I'm like, what am I freaking, I'm so sorry. Pink. 4 are pink. I do have pink here. Pink, 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 pink. And we've got 4 red. Oh my goodness, you can tell it's Monday morning. Oof. All right, and then finally, our phenotype frequency of white is 0. Our allele frequencies. Okay, so we have 8 individuals, but each individual has 2 alleles. So the total alleles is 16. So that's 8 individuals times 2 alleles each. And then we have to count each individual allele, whether it's a big R or a small r. So here we have 2, 4, 5, 6, 8, 9, 10, 12. We have 12 big R's. 12 of 16 is 0.75. And we have 1, 2, 3, 4 of 16, which is a frequency of 0.25 little r. Those are allele frequencies. We can predict the genotypes and the phenotypes of the second generation using these numbers. If a male gamete meets a female gamete, they will make a new flower whose genotype is a combination of both parents. The laws of probability say that the chance of a big R male meeting a big R female is a product of their allele frequencies in the gene pool. So again, we take our allele frequencies if we're going to predict what the probability of having a homozygous dominant in the second generation, it's going to be a product of their allele frequencies. So if we have the frequency of big R times the frequency of big R, so one of them is from the male gamete, one of them is from the female gamete, that will be the frequency of a homozygous dominant second generation individual. So that is 0.75 times 0.75, which is equal to 0.5625. So what that means is that for F2, the second generation, we have a 56.25% of having a homozygous dominant individual. If we want to predict the frequency of a homozygous recessive individual, we take the product of the allele frequencies for the recessive allele from the first generation. So the allele frequency for little r for the first generation was 0.25. So we take 0.25 times 0.25, and we get for F2 a 6.25% chance of having a homozygous recessive individual. The frequencies of all expected types, of course, must be equal to 1. So if we want to determine what the frequency of a heterozygous individual, we can either do the frequency of big R times the frequency of little r, or we can just do 1 minus the frequency of the homozygous dominant minus the frequency of the homozygous recessive. So we take all of that together and we get left over 0.375, or a 37.5% chance of having a heterozygous individual in F2. The last topic for section 1 is kind of related to the topic, or the main title of this section, which is genetic equilibrium. Now, what happens to allele frequencies over generations? These two scientists are named Hardy and Weinberg. They showed that allele frequencies tend to remain the same unless acted on by outside influences. So genetic equilibrium is based on a set of assumptions of a population that is not evolving. So basically what they're saying is a population is going to stay the same unless there's outside influences that act upon that population, kind of like physics. What is that? Newton number 3, something like that. An object at rest will tend to stay at rest unless acted upon by an outside force, whatever. Anyway, the same thing applies for genetic equilibrium. So a population will not evolve unless there's some kind of outside force that acts upon it. If a population is not evolving, its gene pool, therefore its allele frequency, is staying the same. So Hardy and Weinberg came up with 5 assumptions that would apply to a population which has achieved genetic equilibrium. And that is a population which is not evolving, a population that is not being acted upon by these outside forces. Of course, in the next section, we're going to learn that most of these assumptions they don't hold true, which is why populations do evolve. But they wanted to come up with how could a population maintain its allele frequency and not evolve? So assumption number 1, there are no net mutations. So allele frequency is not going to change because of a mutation. Of course, we know that mutations do occur, but like I said, I'll talk about that in the next video on Wednesday. Assumption number 2, individuals neither enter nor leave the population. Of course, we know that's not true as well. Number 3, the population is infinitely large. This is probably the most unrealistic of the assumptions of having an infinitely large population. Of course, we know that does not hold true because every population has a maximum carrying capacity. Number 4, individuals mate randomly. While some species this might apply to just based on the availability of mates, but especially in the animal kingdom, it's usually not random. And finally, number 5, selection, meaning natural selection, does not occur. So these 5 assumptions must hold true in order to classify a population as achieving genetic equilibrium, which means that the gene pool doesn't change, allele frequencies don't change, and the population remains the same. In the next section, I will talk about the disruption of genetic equilibrium and why those 5 assumptions do not hold true.